I am Ria Saha from the Department of Microbiology at St. Joseph's College Autonomous, Bangalore, and I'll be your host today morning. On behalf of the organizing committee, I welcome all of you to the third and final day of Kaleidoscope 3.0. I request all the participants to keep their microphones on mute and camera off, also requesting you to not exit the meeting during the course of the talk, as it may disturb the participants. Kindly post your questions in the chat box and we will present them to the speaker post the talk. We are elated to have our speaker, Dr. ARPIT MISHRA, with us this morning. A very good evening and warm welcome to you, sir. I would like to thank you for accepting our invitation, although it's late evening at Seattle right now. I deem it my privilege to appoint Dr. ARPIT to our audience. He has an illustrious resume, and I hope I do justice in introducing him to you. He describes himself as a non-coding He describes himself as an innovative, self-motivated hybrid genome biologist with a focus in area of non-coding regulatory variants, prioritizing and target identification. He is currently a senior fellow at the Hawkins Lab, Seattle, working on medical genetics and genome science. Dr. ARPIT pursued his undergraduate and postgraduate studies at MS University of Baroda, Gujarat, and completed his doctoral degree from Savitribai Phule Pune University, Pune. He is an expert in experiment designing using next-generation sequencing and is a master of plethora of technical skills like genome editing, chipset, R programming, and Python, to name a few. Sir's expertise extends to managerial endeavors too. Dr. Mishra has also successfully assumed and loved the roles of a teacher and mentor in the past. He has also designed and taught genome editing in disease and treatment to biology major fourth-year undergrad students at University of Washington, Seattle campus in 2019 as a part of the prestigious science teaching experience for postdoc program. He has, to his credit, numerous publications in reputed journals, plentiful citations. We are indeed delighted and gratified to have you, sir. I now invite you to address the audience. Over to you, Dr.
Okay. I will start the presentation.
Sure.
Okay. So, is it visible?
Not yet, sir.
Okay.
Yes, now we can see the slide. It's just coming up. We can see the slide.
Is it visible now?
Yes, sir, it is visible.
So thank you very much for this wonderful program where and giving me the opportunity to talk to your students yeah, it's good outside the research work. I like to interact with students, so I really enjoy this. So hopefully I would be able to do justice to the program, like what you people have organized. And yeah, so I will just start. And so you might be looking at this, why DNA looping and what is the DNA editing? So in the genome, consider DNA is like a long thread. And when there is a long thread and there is a small space, you basically make a loop so that DNA can be accommodated inside the cell. Okay, so that's why the looping, and this kind of looping kind of governs that different type of gene regulation, different type of gene expression in different kinds of cells, which we will discuss through the presentation. And why the editing? So sometimes in the disease, this DNA looping, which is basically a DNA regulatory, gene expression regulatory event, gets disrupted. And like with DNA editing tools, we can basically regain those lost loop, or we create a loop which can be helpful for like normal normal cellular function. So that's why DNA looping and disease. So my talk is basically divided into two parts, because I know like I wasn't having clear idea like who will be the participant, like, and like what is everyone's understanding about this area, basically area of like 3D genome and variants, and how disease variants, so GWAS association studies. So at first part, I would try to bring everyone to the common ground, where I will introduce some common terminologies in the field we commonly use, like genome sequencing, what is genome-wide association study, what are like different types of genome sequences, and then there are like coding genome versus non-coding genome, what happens in the coding genome, what is regulatory genome, and like how we are different from each other, like what are the variation which basically governs the differences among like our response to disease, our response to different environment. And so I would somewhat also introduce this compaction problem and epigenetics, which is basically like which is other than gene, like which is outside the gene, like or on top of the gene. So that's like epigenetics. Many of you already know this thing, but just so that everyone are on the same table, I will still go through this, and then I will introduce one interesting regulatory thing which is called enhancer. Basically, enhancer are a part of non-coding regulatory sequences which helps in the gene, helps gene to express. So, and in the second part, using this all the information, I will try to tell you the story about one autoimmune disease which is recently published by one group from UK. And like I thought right now is a good, so like since using these approaches you can study anything like cancer, you can study any disease like digestive tract disorder or type diabetes, different types of diabetes. But I like with current pandemic going on, and which is one of the main reason of the pandemic is autoimmune responses. So like when body is giving response to the virus, and in turn which leads to the lethal response, and like people develop AIDS kind of syndrome. So I will tell you a story about autoimmune disease which is not COVID, but which is colitis, which is basically a inflammatory bowel disorder. But that would be very interesting story. So what will you learn from this talk, or like what I plan to explain? So one, how this recent development in the field of genomics and genome editing is leading us towards the area of like genomic medicine, where like depending on, and how this genomic medicine, like how this new information will help us identify the novel target genes, novel drug discovery potential genes, that kind of things, and how different genomic as well as epigenomic information, when we combine in a one model, how it can help us gain the better understanding of the diseases. So let's move on to that part. So recently, in last June, like very recently, 26th June, was the 20th anniversary of first draft of the human genome. So basically, first draft was initially announced on June 26th, and then it was fully finished at 2003. So what does mean? What do we understand by first draft? First draft is basically like we have the blueprint of the cell for the human cell. We have the sequence information, and like many of you, I kind of assume like most of you must be aware of this recent advances in this genome editing field, which is mainly driven by CRISPR-based tools. And if you are not aware, I will briefly go through it. But basically, CRISPR is just a tool where you can modify the genome. So, like in 2000, we got the capability to read the genome, and in around 2013, and like even before that, like there are some other technology which allowed us to even write the genome. So basically now we can actually read it, and at the same time we can also write it. So this kind of opens up the like novel avenue in case of genetic disorders. So till now, what used to happen in genetic disorder, like if you can sequence the genome, you can know that, okay, this person is a carrier, or this person has a mutated gene, so he will have a genetic disorder. But there are not lots of treatment options available. But like this writing kind of opened that novel avenue that like we can actually have a gene therapy for, and many people, like many of this therapy, like CAR T cell based therapy for cancer, like which are genetically encoded, as well as Duchenne muscular dystrophy, are already in like advanced stage. And many other, like for eye disorder, there is one therapy which is in like a third phase of clinical trial. So yeah, there is lots of novel avenues open. So remember, we got the capability to read the genome and write the genome. Okay, so like first of all, when you sequence the genome, what generally we do in a biology, we ask that how similar we are when we compare to other organisms. So when we looked at this our genome, and this wasn't very surprising, because if you have studied the evolution, and if you have studied that like how branching happens, like plant separates, then animal, and then from animal, like later on, like there are insecta, then there are mammalian, and this group separates. So like when we look at the similarity, and this was very like for chimpanzee, we have like almost 98% genome, which is very similar. And with mouse, we have 92%, which was very similar. So this is very interesting, like specifically in recent time when like we all have, like in world, it's going on, like how we are different, like this different social issues going on that basis. But technically at genomic level, we are almost very similar. And like, so this, like even we are similar at 98% with the chimpanzee, but like if you compare to like other human, we are almost 99% similar. So how this similarity, how like we are this similar and still we have lots of variation in like our phenotype, in our susceptibility to different disease. So that's the very interesting part, which we will move on. So basically, when we sequence the genome, there are 3 billion bases in the genome. And out of this, only 2% of the genome is the coding region. Coding region is basically the one which codes for some kind of protein or some kind of mRNA. And for 98% of the genome is the non-coding region. Initially also thought as a junk DNA, which is actually not correct. Like it's basically, some people refer it as a dark DNA and many different terms. But like, I just want to make aware. So like there are basically 3 types of genome sequences. So someone asks you, what is the genome sequence? So you can say that one is whole genome sequence. When you have like lots of money, like initially first few genome sequences took almost like $2 million. And now you can do the whole genome sequence in under $300. So those are that's how the technology is developing and how fast things are moving. But because most of the time, if you are studying some disease, you want to see like what are the protein changes, what are the mRNA changes. So instead of doing this 98% of genome sequencing, you just do 2% of the genome sequencing. And that's called as exome sequencing. And now still, like sometimes, if you see, because we have 99.99% genome similarity. So if we want to see like how one population is different, have a different susceptibility to some disease or some like height or weight or something, we can technically do just 0.03% of genome sequences and can get the idea that what are the variants or what are these small differences which is driving this phenotypically large differences. So like, so these are also called as SNPs or single nucleotide polymorphisms, and which are generally present every 1,000 base pair. So like if you compare the two genome, like your genome and my genome, at first they will look very similar. But if you go into the details, at every 1,000 base pair, there will be some variation which will be there. Like, so in my case, it is possible that I have at this position, I have T, then you might have A or some other variants. And like that is also stratified in the population. Like some people have like 50% population will have A, 50% will have T, or sometimes it is 95 to 5, that kind of ratio. But this is called genotyping, which is basically only sequencing or only getting the information about these variants, which kind of drives the different disease susceptibility. So when we look at this genome, just see that number. It's like 3 billion bases. And the first question, what anyone will ask, that like how long this would be, how these 3 billion bases can be accommodated inside the cell. So for that, we will look into these two main questions. So how to accommodate this 3 billion bases inside the cell, which is like really small. It's like 2 micrometer cube. And this 3 billion bases is approximately 6 feet DNA. So, and this is not just like we want to just like do some kind of origami and just fold the DNA so that it's there inside the cell. We also, like cell also wants it to be active at a time. Like it wants to have access of certain genes to the transcriptional machinery. So this becomes a very, very interesting problem in case of the DNA compaction problem. And the second is what is the function of non-coding elements? So can you see this 98%, this green colored is a non-coding element. But once we have this DNA sequence in 2003, many of these non-coding elements function was not clear. Like we can read the information, but we could not understand what is there, like what this DNA is doing, like what this sequence is doing. So for that, there was another effort started after human genome sequencing effort that's called Encyclopedia of DNA. Like they tried to study other feature other than the just plain sequences and tried to classify the DNA, non-coding DNA in some functional classes, which we will look into the second part. So first look at the 3D genome organization. So I think everyone knows this guy, Charles Darwin, and he was approximately 6 feet long. And basically when you think of a DNA inside the cell, this is one of the somewhat similar analogy you can give, that you want to accommodate this guy in this zoomed out salt grain, and still you want this guy to be functional. So that's the exact scenario for DNA packaging. You want to package is very efficiently, but you want to also allow the DNA to be functional. You want to allow some part of the genome to be active, some part to be inactive. So for that, they took this very simple methodology. Like Cell adapted to this methodology. Like if you have like long thread and you want to compact it, you can use like some kind of ball or some kind of bead, and like you start putting those thread around the bead, and that way this length of the DNA will reduce, but this information is still accessible by the transcriptional machinery. So this is called accessible DNA. But together, this histone protein complex and the DNA is called chromatin. And this chromatin has like how this remains accessible. So there is some kind of epigenetic code going on here that histone tails have some kind of modification. So consider this as like this was the DNA sequence, and DNA sequence on the top of the sequence, there is some kind of information here on the histone tail, which tells that if this DNA needs to be open or this, and like if that DNA needs to be closed, all these beads will come together and form this kind of heterochromatin, which will become DNA inaccessible. So like in which scenario you will need this? So let's say you are a neuronal cell. So you don't want to express the hemoglobin. So you, in the neuronal cell, this hemoglobin will be heterochromatinized, and the other neuronal related proteins will be euchromatinized. So like will be accessible and this will be inaccessible. So that way it kind of have a delicate balance. And generally, like you have what cell try to do, it try to put this. So these are different chromosomes and different chromosomal territories. So like which is compacted like at this level, and cell try to kind of compartmentalize. So if cell wants certain things to be active, it will try to keep it inside the some related area. So like all the transcriptional machinery can easily access. And if it wants to switch off that region, it will try to put it near the nuclear membrane. So there are some exceptions, but like this is the general principle. So basically, like you will have like multiple loops, and there is like smaller loops of the loops. So this way you basically compacted the DNA, but at the same time you also allowed the certain type of gene expression access, and you also allowed some regulatory interaction to happen. So this was about this is like epigenetics help in the DNA compaction in this manner. So now let's look at what is there in the non-coding genome, or sometimes referred as dark DNA. So for this, after the human genome was sequenced, there was this consortium started, which is called Encyclopedia of DNA Elements. So many group of scientists came together and they started developing some technique to further classify this DNA element based on their some chemical features. So like this is one method where this is like a contact tracing. So like if this region of DNA in 3D came near another B region, then in this kind of methods, A and B will be cross-linked together. So you will know that in linear distance, A and B might be 10 kb apart, but in physical distance, in 3D distance, they will be very nearby, and there might be some regulatory interaction happening there. So similarly, like if you want to have DNA accessible or readable by the machinery, you want to keep it as open as possible. So like these are the methods which tells you if the region we are looking at is open or closed. So like if there is a DNase or ataxic signal, this is one of the method. Like I will not go in detail into method. You can ask me later on, but just for the understanding, this is one of the DNase method where if the DNA is accessible, this region will be chewed by the DNA and that signal will not be there. So depending on that signal, we can say that which region is open and which region is closely compacted. Similarly, there is another method which can tell you that like what are the proteins present at that particular loci we are looking at, or what are the histone modification which is like happening at the histone tail using this chip check type of method. So the ultimate idea, you can further give a different feature to normal sequence of DNA. So like when we sequenced, we had just added this information that like DNA is like ATGC, some type of different combination. But after this project started, basically you can divide this your same DNA. So this is the DNA like position chromosome to like this different position, depending on the signal they have. Like if they have H3K for ME1, so like this signal is classified two major categories: histone modification and open chromatin. Open chromatin indicates that this region are active inside the DNA, which is still compacted inside the cell. And histone modification also somehow correlated with, but like let's say this H3K for monomethylation mark. So there it is called epigenetic code, which I will come later on for some specific region of interest. And this indicates that there is some kind of enhancer. So, and this indicates there is some kind of transcriptional going on for this gene. And similarly, like different, like this is promoter, there is this transcription going on. So like different histone modification and different technique will tell you or give you a different feature for that particular region of your interest. Okay, so now we know there is a coding region which forms the transcript, and there is a non-coding region, promoter or long-range regulatory element, sometimes referred as enhancer, repressor, silencer, or insulators. So do this regulatory region or non-coding region interact with the coding region? So for that, I always had, like when I was in BSc, I always had curiosity, like if all the cells in my body have the same DNA, why particular cells becomes like red blood cell, why some cells become intestinal cells, why some becomes neuron? So that answer somewhere lies in this information. Like when we look at this information, but like the interesting part here, so just think of like how imaginative the scientists were. So this guy in 1950, his name is Conrad Weddington. He somehow, like even like at that time, DNA was also not discovered. Like not many people had like DNA sequence idea, no DNA structure idea. But he somehow hypothesized that cell are like when they are undifferentiated or like a stem cell, it's like on the top of one hill, and when cell starts going down, it can go into different paths depending on what are the gene switch on, like what are the genes which are switched on particular. And these are like most of the time this path are like unidirectional, you can only go once, but there are always exceptions in biology. But like, so just think of imagination that this guy came up with this nice idea which is like very relevant today after having all this complex information about DNA. So what basically drives this? These are like a different master regulator. Like for every cell, there will be some master transcription factor, which and there will not be just one, but there is a combination of this transcription factor. So can you think of like if you have just promoter where the transcription factor can bind, you cannot create a multiple combination of this event. So this non-coding region have some kind of regulatory switches. Here we will look at the enhancer, which basically provides another surface, and because they are coming together in 3D, that facilitates some kind of a specific gene expression pattern. Let's say in epithelial cell, there is some kind of enhancer, like which are cells type specific. They bring some particular transcription factor nearby this particular promoter and then allow the expression of gene. So that way, having the same DNA, but different enhancer usage and different transcription factor usage gives rise to different cell fate. So just to give you further idea about this. So let's say there is this gene which is required in all the different cell types, but they are required in different level. So let's say this in the skin cell, this gene is required at the basal level. So here the enhancer will not be used. But in the liver cell, this gene is required at high level. So there will be this another accessory enhancer which will recruit the activator and thus increase the speed of this production of this mRNA and ultimately protein. And in brain cells, you want to actually have minimal or very low level of expression. So at the same position, or sometimes you recruit some another, like instead of enhancer, you recruit a silencer, which basically brings some repressor element and reduces the gene expression. So, okay, now we looked at the enhancers. But how many different types of enhancer and where it can be? If, let's say this is my promoter, the enhancer can be downstream to the promoter. So like this is my gene start. It can be upstream to the promoter. So these are like some upstream enhancer or proximal promoter enhancer. So like basically you can classify your enhancer in different category: upstream enhancer, downstream enhancer, or a distal enhancer, which is basically like hundreds of kb away. And technically, when you sequence the DNA, they will be like far, far away. But like because of 3D organization, these things come together and basically create some kind of a modular switch. So only certain type of gene expression are allowed. Okay, now we are we have now this genetic polymorphism and disease. We look, so like when we look at the two images, like when we look at the two genome sequences, they are basically very similar. Like they will look like this, and but like when you want to look at how different they are from each other, there are only like these small variants. So I will like refer to those questions afterwards. So yeah, but I will come back to that question. So like this enhancer or like this genome will vary at this position of a this variant. So technically every cell get two chromosome, one paternal and one maternal, and these are called homologous chromosome. And at each locus, one part will be from maternal side, one part will be paternal side. So like if this is the variant, one is coming from father, one is coming from mother, and if these variants are similar, they are called homozygous. If they are different, like one father has the dominant and mother has the recessive or vice versa, these are called heterozygous variants, and they kind of drive this different type of enhancer, like how enhancer can function differently. So just to clarify you, what is this SNP? So let's say there are 6 individuals, and we are looking at this particular position in the genome, which is basically we found from the genotype information that this guy got this T and C information here. So it is heterozygous. This guy also got T and C. This guy got CC, so this become homozygous. This guy got CT, this is heterozygous, and TT. Like so that way, like every so that's why this is called single nucleotide polymorphism, because in the population there will be like different people will have in the same location will have some kind of a stratification, like some like disease causing variant will be like less prevalent, so like they will be just like 5% or even less than that, or while other variants would be more prevalent, like 50-50 or 60-40 depending on the population. So these are the information. So basically like when we look at the DNA, like you might have seen in during a different crime movies, or like they do DNA sequencing, like and they tell, oh, this is from this guy, this is from this, but like think of it, it's 99.99% similar DNA. So these are the variants which basically tells you that which variant combination is coming from a different person. So we differ at the variant. So now I told you that like if we have this technology to do the only variant sequencing, which is just 0.03% of the genome, so people thought of this experiment. We already had this idea that like different population have a different susceptibility to disease. Like there are some cases, like let's say we take one example of diabetes. There are people, so like scientists took lots of diabetic people, and at the same time they took the age matched, sex matched control, and they did this genotype sequencing, and they found that like for cases there are some variants which are highly associated with only cases, and they are not present in the control. So basically this gives you a possible target gene that drives or target a variant which drives the disease. But this, so there is one another problem. Now, as I told, 98% of the genome is non-coding. So what happens when you look at this variant? Most of the time, more than 90% of variant are in the non-coding region, and out of that 60% reside in the enhancer type of region. So how do we get this list? Like, do we need to do this study every time? No, people have already done it, and like there are lots and lots of GWAS studies are available. So like, let's say you are a drug company and you are looking for, you are working in cardiovascular disease. From this GWAS catalog, you can download all this variant database, and then after that, what will you do? There are multiple paths, but like for today's story, I will tell you that how we can use this variant information, disease associated variant information, to understand the disease mechanism. So let's say you got some GWAS hits. this will be in two categories. One is coding region, one is non-coding region. If this GWAS hits in the coding region, let's say this RS334. So these variants are generally called as RS, then some number. So RS334 is AA, which is a normal, AT, which is a carrier for the sickle cell. So like if you have, this is in the coding region. So you can clearly see that this A leads to glutamine formation and the hemoglobin is normal. But if there is a T, and like if both the chromosome have T, then they will have a dysfunctional hemoglobin, which leads to sickle cell disease. So this was very simple puzzle. But now think of a scenario where you have a non-coding region variant, and which is very prevalent. Like in 90% cases, your variants are in the non-coding region. So how do you identify the mechanism? Like what disease this will be, how this is causing disease, or like what gene it is regulating. So you have to become some kind of like a genomic Sherlock Holmes to try to solve this mystery, that like what this non-coding region, which is not lying in any of the gene, what gene it is regulating. So sometimes initially people thought, okay, let's look at the next gene. It should be the regulating the next nearest neighbor gene. But like with this idea of 3D genome, your like linear neighbor might not be the same as your 3D architecture neighbor. So you also want to look at those type of information. So that's so to just to give you the flavor of like how this enhancer variants are causing the disease. So this is like one enhancer variant at this particular locus CMO. So this in the wild type, this regulates this PAX6 locus. But if this becomes G2T, then there is a disease called aniridia, which leads to this disease. Then there are some other, because in the genome, variant is one type, then there is sometimes what happens, by mutation, this enhancer got deleted. So they got this kind of Peary-Robin sequence. And then there are sometimes C2T happened here. So this ZRS is regulating SSH at particular level, because consider this enhancer are like a fine tuning agent. So you don't want to have like lots of enhancer contact also. You want to like have at some level, or like you don't want it to become very strong enhancer also. But because of this mutation, this becomes strong enhancer and you get this polydactyly. Like just to give this, Hrithik Roshan has this polydactyly, but I don't know if it is driven by this. And there is like similar example where this different enhancer got duplicated in the genome, and then again you have this kind of disease. So basically enhancer variants can give rise to disease. So that's the story. Now let's come back to the story about regulatory variant in autoimmune diseases. So think of this, autoimmune diseases are the cost-wise in NIH estimated that autoimmune diseases have higher cost than even cancer care, because this generally affects the younger population. There is like lots of cost in caring about the autoimmune diseases, and it kind of affects women more frequently than the men. And these are some common autoimmune diseases like asthma, type 1 diabetes. But so people already did lots of GWAS study related to different autoimmune diseases. So just to brief, GWAS is this study where you have the list of variants related to specific disease. So they have lots of list of different autoimmune disease, and they have already have list of variants affecting or like predisposing person to the IBD or other autoimmune disease. So today we will look at this particular disease, which is called inflammatory bowel disease, or commonly called as colitis. So what happens? So this is the healthy colon, and this is the colitis or IBD colon. So you can see person will look very normal. These are not like that injury which will like clearly visible at first, but they will be suffering a lot because of this kind of damage going on. And this is like their own immune cells are damaging their colon. So this group, Rahul Roy Chaudhary and Gosia Trinka from UK, they actually use this very intricate mechanism to identify this non-coding variant which is also called as 11q13.5, to identify how it is causing or how it is promoting the suppression of colitis in Treg cells. So okay, now let's look what is Treg cells. So in immune system, there are different types of cells. There are like T regulatory cells, then there are like T effector cells, CD4, CD8 cells. There are repertoire of the cells, and cell goes through, like this cell goes to multiple selection so that it doesn't start recognizing its own cells as the pathogen and doesn't start killing it. So this Treg cells basically acts as a brake. So if there is some T effector cell which is killing the own cell, Treg cell somehow, or like this is one of the mechanism. The Treg cell produces this transmembrane protein called GARP, and which basically sequesters the inactive TGF beta and activate it and targets this T effector cell, so that this T effector cells which are killing this intestine, lungs, or skin, depending on different autoimmune disease, that will be repressed. So as I told you, this question becomes very easy if this is in the coding region. But here, this scenario, this risk locus is in the non-coding region. So how to identify what the gene it is regulating or how it is regulating? And to understand this, we took a different, like in this paper, they took a different approaches. So first they asked that like this variant, although it is in non-coding region, is lying over some kind of regulatory sequences, and how we got this information about regulatory information based on the that ENCODE data. So like if there is a H3K for monomethylation in histone tail, that indicates these are lying on the promotory enhancer region, or like H3K27 acetylation. So second thing, now you want to confirm your finding. So you want to also do your experiment, some kind of related disease model. So they identified that in the mouse, similar regulatory circuits are conserved. So they used this mouse in the study. Next, they asked, okay, we found that this is overlapping. What are their 3D neighborhoods? So using one of the method called HiChip, they identified that in 3D, what genes these are interacting. So, okay, now these are the observational data, which is already there. So next, if they identify that this is the actual target gene, if you perturb this variant, your target gene should also get changed. So using CRISPR, they basically did this perturbation type of experiment. And in the third and the last, which is they use some kind of EQTL. So what is EQTL? Is you basically like so this is like naturally occurring experiment. So if there is a patient, you get like 3 type of patient, which are heterozygous, homozygous for AA, and homozygous for GG, or which is a risk allele. So you can say that like, yes, your target gene is somehow correlated with this genotypic information. So that is called EQTL. So we'll quickly go through this. So we asked, so in this paper, they asked this, okay, well, this is the variant. Is that variant overlapping with some kind of enhancer-like feature? So they looked at the human chromosome 11, and they had this peak signal that this, yes, this is the enhancer. So they found that this is overlapping with the enhancer. And once this is overlapping with the enhancer, so what it might be happening, that because of this variant, it might be recruiting some kind of other transcription factor to the possible target gene. So yeah, so our first criteria is satisfied that yes, this variant is indeed a regulatory variant because it is overlapping with the regulators. So next, to look at the animal model, like where you want to do study, is the genetic and epigenetic feature conserved? So they looked for human and mouse, and they found, yes, this circuit is very much conserved, not just at the genic level, but at epigenetic level also. Okay, so you can see that in this mouse cells, this enhancer is conserved, this gene is conserved, and the enhancer marks are still also conserved. And that too only in Treg cells, because this enhancer is functional in Treg cell and not in other T conventional cells. So this kind of satisfied the second, that like, yes, the model they are using kind of, okay, I will get back to the question. So next, okay, now we have this two information fulfilled. Overlaps with regulatory region. Animal model is conserved with this kind of regulatory circuits. What are the 3D neighbors? So for that, they use some kind of high chip information. And at first time, they asked that like when I look at this position, which is the RC32 promoter, which is basically a target gene they are looking at, or like they kind of identified based on this high chip interaction. And they found that yes, indeed, there is an interaction happening between this and this. So this is the this enhancer variant, and this is the target gene. So from 3D variant also, they could assign that yes, this variant, which is disease susceptibility variant, is somehow interacting with this target gene. So next, so till now they did whatever like observational information is available. So next they wanted to like perturb this particular variant location and see that if that perturbing that location actually gives some kind of disease phenotype or some kind of change in that particular gene expression. So this is like, I quickly go through this CRISPR, how you design a CRISPR experiment. So sorry, like, am I running out of time or anything? Okay.
No, doctor, you can continue. Not a problem.
Okay. So let's say this is your region of interest. You want to delete it or you want to disrupt it. So using CRISPR, you can do multiple things. You basically, if you use just one guide RNA, so CRISPR is basically a molecular scissor, and given the guide RNA, whatever guide RNA you provide, it will go and try to cleave that region. And now with the CRISPR, there are many variants are also available. So these are the CRISPR-A or CRISPR-I variant, where cutting part is mutated, but the localization part is still there. So this can go and activate this region. So this can give signal that like recruit more activator. So like if this is the target gene, then this target gene should go up. And this is opposite, like recruit a more repressor. So if this is the true target gene, this will go down. And you can also create like a large deletion. Like if you use two functional CRISPR in that region, you can create a large deletion. So they basically did two things. They use this large deletion and then they use CRISPR-A activation. Okay, so when they deleted this enhancer region or risk loci, and then, so always consider this kind of autoimmune diseases are not just like if you have genetic predisposition, it will always happen. There should be some environmental trigger. So they kind of triggered this disease in presence of this DSS. So the DSS is one compound which basically induces the colitis-like phenotype. And you can clearly see this body mass suddenly start reducing for the enhancer knockout when compared to wild type. So like you can see, this is the size of the colon in the mouse where enhancer was knocked out, and this is the wild type. So this is clearly visible that this enhancer is somehow, deletion of this enhancer is somehow causing the disease. They also looked at the histopathology, and they can clearly see that like histopathology score is very high for enhancer deletion, because like these are the damage happening which are induced by like its own cells, like T effector cells, which are basically damaging it. So this kind of confirmed that disrupted enhancer leads to the disease. So like enhancer was somehow protecting against the disease. Okay, now next they ask, when they delete the enhancer, what is the gene which is upregulated or which is different compared to the wild type? So this is the gene like which is expected that this LRRC32, which codes for this particular GARP protein. This was clearly difference between wild type versus enhancer knockout. And in the GARP, so this GARP is basically a 72 kilodalton protein, And Treg cell is like a immune suppressor. So if there is autoimmunity going on, this effector protein, which is basically killing its own colon, this Treg cell will repress this. So thus there will be no killing. So what happens when you delete the enhancer? Your level of GARP reduces drastically in activated and rested Treg, in both the type of Treg. But this enhancer deletion has no effect on different types of cells. So like that indicates that this enhancer is only functional in this kind of Treg cells and not in like a lung endothelial cell or platelet cell, which are also expressing this thing. So next, okay, we got that this enhancer has a mutation, this enhancer has this particular variant, all the information we have. But now, next obvious thing, like how this variant is affecting, how this is causing the disease. So when they looked at the genome sequence, we find that this enhancer, nearby the enhancer, there are two transcription factor binding motifs, one STAT5 and NF-kappa beta. And using the chipset type of methodology, they clearly show that there is an enrichment of STAT5 in functional enhancer location, which basically recruits STAT5 to this GARP promoter, and similarly it recruits the NF-kappa beta at the GARP promoter to like enhance the expression of GARP gene, which you can see clearly here that when there is a wild type, there is a GARP expression. This axis leads to GARP expression. But when there is a deletion of enhancer, there is no GARP expression. Same here. These are like a different activating condition, like because immune cells are also get activated by like different methodologies. So one time it was activated using IL-2, other time in the cell it might be activated using CD328 pathway. So, but you can clearly see, at least specifically here, when it is activated by IL-2, there is no expression, not at all a single expression of or like GARP2. So thus Treg cell function would be compromised here, and which will lead to the disease. Okay, so now we come to this another part of the story. So when I told you like at 1000 base pair locus, there are multiple variants. So how do you like differentiate between what variants? So like here, it's not just a one variant. These red dots are a variant, which we are currently investigating is RS11236, this variant. But there are other variants which are also enriched in this location and at this enhancer locus. So how to pinpoint the function that like which enhancer is actually or like which variant is causing the disease? So for this pinpointing the function, they use this another type of CRISPR experiment where instead of using the deletion approach, they basically activated this particular loci. And once they have activated this loci, so they have like different variants because these are the variants in that particular loci. They designed a guide RNA, and this guide RNA also has the GFP in it. So you make sure that yeah, there is a guide RNA expressing. So there is your CRISPR activator is going to that location. So for all these three examples, like guide RNA 9, guide RNA 11, and guide RNA 13, you can see that there is indeed guide RNA expressing, but do we see increase in GARP? This only happens when we targeted the G11, which is this particular variant which they have identified. So this kind of fine maps, like among the list of variant, which variant specifically caused the disease. So, and this is the last, which is like a naturally available experiment, because when they have patient with like all these different genotypes, CC, which is like protecting, CA, which is heterozygous, and A, which is a risk. In those cases, they can clearly see that this enhancer is having a higher enhancer-like signal compared to A. So somehow enhancer-like signal is affected in this A phenotype, but this effect is not there in T conventional cell. And similarly with the GARP expression, that CC is somehow protecting, so there is more GARP expression, there is CC, when there is CC compared to when there is AA or CA. So this kind of confirms this our like finding, and they kind of attach this non-coding variant to possible target gene using this different approaches. So this kind of tells you like one complete story that how a variant in non-coding region can cause a disease. So yeah, I would just like to thank my lab, and I would like to thank the organizer and this one of the collaborator, and special thanks like because he's my MSc teacher whom we lost this year, that he actually introduced us, like where Shalmali was also in the same batch, to this chromatin and 3D genome architecture. Yeah, so I will now come back to the question. And sorry, I got bit.
Not a problem, Dr. Arpit. I'm sure all the participants have thoroughly enjoyed your session. I would like to thank you on behalf of all of us for this beautiful story that you weaved using epigenetics and 3D genome and GWAS and autoimmunity as their main characters. I'm sure we have all got a lot of concepts to take home, and I would speak on behalf of all the participants. Also, thank you for introducing us to the future that medical science and medicine and disease management would take. So we will proceed to the questions. We have quite a few questions.
So
I would request you to
read the question or whatever.
Sorry, I'm sorry.
Read the question, or you want to, like, I can.
As you wish, I can read out the question to you and you could answer.
Yeah.
Can I please request you to stop your screen sharing so that the audience can see you full screen?
Yeah.
Thank you so much. Okay, so we have two questions from Dr. Madhvi Kaji. Her first question is, can mutations and variants in enhancers influence human disease? If so, how do they?
I think this is very important question, and like all this study and like most of the like our group and many other groups are working in this direction, because now that is obvious thing, like, and also from the normal overlap analysis we find that most of the time these variants are like 60% of the time variants are in the enhancer. So yes, indeed they affect the disease. So I actually showed one figure, like I can go back to the presentation, but like if you remember, there is this loop happening and this A to T happened in the enhancer, and thus this aniridia disease was causing the aniridia disease. So yeah, this is a very relevant question, and that is very common, like one of the way ahead, like to find the novel disease mechanism. And
It has posed another question. I think, does having IBS make you immunocompromised?
Yeah, so I wouldn't like comment on this part because I am like not expert on IBS part. I'm more into like gene regulatory events part, but I haven't come across any literature when I go through the autoimmune disease where IBS makes one immunocompromised. So I'm not sure about, yeah.
Okay. Then we move on to the next question that has been posed by Dr. Syed Wajid. And he asks, what techniques are used to study and assign non-coding regions that regulate genes?
So yeah, this is very interesting question, and which we are commonly using it. So should I go back to the presentation or?
Sure, sir, whatever you're comfortable with.
Yeah, okay, then I will start the. Is it visible?
Yes, sir, it is.
Yeah, so this is very relevant question, and how we can narrow down to this disease related, like disease causing variant. So like, let's say I'm studying the autoimmune disease, rheumatoid arthritis. Basically, I look at this GWAS catalog for rheumatoid arthritis disease. So here there is a list of rheumatoid arthritis, and these are these small dots These are basically a chromosome, and each dot represent there is a disease associated variant at that particular location. So now I have a list of variants for that particular disease, and next I ask, depending on the disease, like I know for this disease, this generally happens via T cell. So I ask that, like, what are the enhancer-like marks in the T cell? So I basically ask this question, like in the T cell, specific T cell type, how many STK4 monomethylation peaks are there, and how many of these peaks are overlapping with the variants? So that gives me enhancer variants.
Okay. Moving on to the next question. It is from Mr. Rohit Gaura. Can we use this gene therapy against a particular disease? Is this as effective as chemotherapy or like immunotherapy or any other treatment in curing the disease?
So this is also a very good question, and like very upcoming field. There is this disease together are called enhanceropathies. So it's like because of disease with mediated by enhancer. And yes, there are lots of efforts, but as usual, like these study are in like very early days. So like if you ask, can we treat cancer in mouse? Yes, we can using this. But for like to go this into the human, this will take time. But like, yeah, we are very much in towards that direction.
Thank you, sir. Then we have Gauri Sharma asking, why is there a time lag in the development of symptoms of autoimmune diseases when the mutation is present since birth?
So again, like we have to go back. Like if you remember in this experiment also, they had the mutation, they had this enhancer deleted, but still when there is no stimulating agent, there is no disease. So basically, specifically with epigenetics, it's not just genome, but it's also environment influences. So some people who have like chances, who are like predisposed to the disease, but because there is like proper environment, proper diet, like let's say for diabetes, I talk about diabetes, like which has lots of epigenetic role in it. So like if you do regular exercise, like there are two twins and one is doing regular exercise, the other one is not doing, the other one is not developed, other one is develops, and this guy does not, because there is like a gene environment crosstalk happening via epigenome changes. So that is also one of the important factor, and that's why there are lags. So you need to have some kind of a stimulus.
Interesting. Then we have Anushka Raghubansi asking, what is the easiest technique to detect if a non-coding gene controls or causes a genetic disorder?
So I think this technique wise, I would not say something is easiest. Like, so like you will not believe, like when specifically when you go for making drug, you will not believe like only this, like, okay, this is the 3D neighbor, so this must be the target. You have to have like combinatorial of techniques to say for sure, like if some pharma company is going to invest in this particular drug target, they want to look for like multiple experiment, and that's why they did in this experiment. So like first they look at like if this variant overlaps the regulatory region, if this is some animal model where which can be recapitulated the disease, there is 3D neighbor, and like once we have this, what happens if you perturb this? So I would say like all the combination is required, but like to the start with, I would say this would be somewhat easier, because sometimes there is chance that like in cell type of your interest, already this enhancer marks like chip data is already available, and GWAS hits are also available. So you can start from here.
Wonderful. The next question is from Dr. Beatrice Sequeira. She's asking, since autoimmune diseases like rheumatoid arthritis, etc., are seen at the later stage in life, can you do editing at that time? Since age also influences immunity, can the cells produce enough protein for correcting the disease? So she has posed two questions. One is, can the editing be done at a later age in life? And since age and immunity are interconnected, can the cells produce enough protein for correcting the disease?
So this is very like somewhat debatable topic, and but like at least for this kind of disease like rheumatoid arthritis, which mainly governed by T cell like T regulatory cell, Th1, Th2 cells. So you are not going to edit the actual genome. Like you are not going to, as long as you are not editing something which will be going to the next generation, the technical difficult, because we have to also consider this editing comes with its own risk of like off targeting and other thing. But like specifically with rheumatoid arthritis, one of our collaborators, they just came up with this CAR T cell kind of therapy where in, like from those T cells are isolated from the same patient and they are edited outside the body. So there are like whatever off target happens only to those particular T cell, and they check it thoroughly before like re-injecting. So now these cells are from the same person and they are edited outside the body. So they are somewhat safe. But again, this homing to that particular site and will they produce enough protein or not? Obviously, yeah, there might be some age-related factors, but there cannot be like one answer. But yeah, this is one of the like very challenging field where like lots of people are trying to add this kind of like specifically like for cancer therapy, CAR T cell is therapies, and mostly because cancer is also somewhat similar, like develops later on the age, and CAR T cell, like some kind of CAR T cells are working really well. So that is kind of hopeful for this kind of disease also.
The next question is from Amrita K. Can germline gene editing cure devastating genetic diseases, and is it regulated?
Sorry, can you repeat?
Can germline gene editing cure devastating genetic diseases, and is it regulated?
Regulated by what? Like enhancer or? Okay, yes.
Sure.
So like, let me give you one example. So there is a very high probability that there is one disease called Duchenne muscular dystrophy, which is mutation in CFTR gene. For that, lots of adenovirus-based therapies are already approved in like phase 2 clinical trial and this. But like obviously with those things also there are other that sometimes you edit this cell, but like this cell also dies. Like there will be replenishment of the new cell. So there should be like a homing, like you want to target a stem cell, which basically like totipotent cell, which basically gives rise to those cell population. So yeah, that is happening. And like at least for 2 disease I remember, there is for aniridia and this DMD, they have made a good progress with respect to gene therapy.
Moving on to the next question from Bala Kumaran KV. Could you please explain about GARP?
Okay, so GARP, like just to simplify all the things and to reduce the complexity, Treg cell provides a site where inactive TGF beta can be activated, and in turn this activated TGF beta represses the T effector cell, which is causing the disease, like which is causing the autoimmune disease. So here the GARP function is to facilitate the inactive TGF beta to become active and repress the T effector cell.
Then we have Vaibhavi M. Is it possible to isolate certain cells and express the silenced gene to make functional proteins?
Yes, so like which is commonly done in the lab, like when you want to make proteins. So if you just clone your gene of interest in a plasmid, which has a eukaryotic promoter. So why those genes are silenced? Because transcription factor for that promoter and enhancer for that promoter are not available in that particular cell type. But if you use some commonly expressed eukaryotic promoter, you can express any protein in any cell.
Then we have Nessa Lobo asking, can CRISPR be used to cure any kind of cancer?
So CRISPR is now at least used for few cancer, like based on this CAR T cell therapy. So yeah, so we can say at least for blood cancers it is used. And one of the so I have my university, so there is one very big cancer research institute called Fred Hutch nearby, where they are routinely doing this, like they are doing the CAR T cell type of therapy. Yeah, so genome editing has a promise for cancer.
Wonderful. Then we have Mamta S asking, are there tools to identify if an enhancer affects a single risk loci and not other important functional loci?
Yes, so this is the tool. This is called fine mapping. So they fine mapped it. So what they did for this risk loci, they wanted to elucidate if this is only the risk loci or other loci is also involved in the function. So they targeted this loci and other loci using different guide RNA with this CRISPR-a. So now, if this loci is actually involved, your GARP expression should go up. So we can see it only goes up when we targeted this particular G11 loci and not in 9 or 13th loci. So yeah, so that is called fine mapping, which is feasible using this type of tool.
We have about 3 more questions. Will it be okay to ask you right now? Yeah. Okay. Yeah. So then we have Lena asking, are all enhancers specific to cell type or they are specific to a particular gene?
This is again a very interesting question, and in enhancer biology, as usual, and in any biology, there are always exceptions. So there are some cell type specific enhancer, which basically drives the cell type specific gene. Some are very gene specific enhancer also. Then there are novel, so which we are finding now that there are some enhancer, group of enhancer together regulating one gene. There are group of gene which are competing for the different enhancer. So there are all the possible scenarios there. But yeah, we can say that there are lots of cell type specific enhancers.
We have Rahila Bashir asking, why are some children not similar to their parents? For example, in physical appearance, like skin color, etc.
Yeah. So again, this if you look at, we can if you want to answer this question, you can take the similar approach. You can go here, search for GWAS catalog for the skin color variants, and then try to taste those, which variant is affecting the melanin production. That might explain, because again, there might be some environmental factor, because this is always a crosstalk between epigenetic and environment. That's why.
Absolutely. Then we have Neha Bhatt posing the question, do the disease enhancer or regulator expression in particular cancers vary? Like one person has more and the same diseased person's genome has less expression.
Yeah, so you can think of this this scenario where due to this disease, and this can happen with any type of cancer also. So there is this ZRS enhancer which is regulating the SSH. Mm-hmm. Now, somehow ZRS enhancer had this mutation C2T, and that becomes very strong. And that's why the dosage of the enhancer to that particular gene increased. So that particular gene has now started expressing more of the SSH. And same thing can happen with any protein oncogene, or if the opposite happens, something like this happens with the tumor suppressor gene, where the mutation led to the disruption of this circuit, then again it can cause cancer. So yeah, enhancer have the relationship with the cancer.
Sandhya Santosh is asking, does alteration in the gut microbiome have an effect on IBD?
Yes, that is a different area of research altogether, and there are lots of people working in that direction. So basically, like gut microbiome is also one kind of environment influence. That like your environment, like gut microbiome environment is different, and that's why your signal, your trigger might be different. So yeah, there are evidence towards that.
That brings us to the last question for the session from Sri Devi. Are all non-coding genes responsible for enhancer and promoters?
I did not understand the question. So like, are all non-coding
genes responsible for enhancer and promoter? I guess she wants to ask whether it's the non-coding areas where enhancers and promoters are present. I
kind of have some idea. So like, these are, let's say, non-coding. That can be multiple things. They can be enhancer, they can be repressor or silencer, they can be insulator also. And this is also like in cell type specific context. So like this thing has a, like, it can become enhancer, but because this is right now closed due to DNA compaction, that is not acting as enhancer. So yeah, so like some enhancer are switched from one cell type to different cell types. So like in one cell type, that region is not enhancer, but in another cell type, it becomes enhancer.
Mm-hmm. Absolutely. Thank you so much, Dr. ARPIT. That was the last question. So I would like to thank you on behalf of all of us at St. Joseph's College, the Department of Microbiology, and all the participants for taking out time. And I know it's pretty late there. It's almost close to 10 o'clock at your end. And for still graciously accepting our invitation in spite of the timing not being so great on your part, and for patiently answering all our questions, and for that insightful presentation and insightful journey into so many new concepts. I'm sure a lot of us have benefited from that. I heartily thank you on behalf of all of us.
I was thoroughly enjoyed, and specifically this question answer session is thoroughly enjoyed. And like, feel free to drop me any question. Like, you can get contact from Salmali, or I am also there on Twitter. So maybe
that's so gracious of you. I'm sure it's appreciated by all.
St. Joseph's College for organizing this kind of events. Yeah, it's wonderful.
It's our pleasure. It's our pleasure, and we are glad to have you, and we definitely look forward to many such collaborations.
Yeah. Thank you.
Thank you so much, Dr. ARPIT. Thank you. On that note, it's a wrap for today's first session, and we will be here back at 5 p.m. for the finale session of Kaleidoscope 3.0. To all the participants, the link for the final section is provided in the same email that you have received, containing the link for this morning's meeting session. I request all the participants to join in by 4:50, and we would be starting sharp at 5 for Dr. Rini's talk. Thank you for your excellent and overwhelming participation and interesting questions, and for joining us this morning, and see you soon. See you at 5 p.m. today. Thank you. Thank you, Dr. ARPIT. Thank you to all the participants and the organizing team members, my colleagues. Have a good day, stay safe, and see you in the evening.
Thank you.
Thank you, sir. With that, I request the participants to sign off, and please join us for the finale session in the evening today. Thank you.
Thank you, Ria.